In this video, we'll be talking about neuroinflammation, which refers to inflammation that occurs in the central nervous system, which includes brain and the spinal cord. So this inflammation could happen due to brain injury or even infection. So let us take some examples. So one of the key disease where neuroinflammation happens is the meningitis. So our brain is protected by a layer known as meninges, and this is how a normal meninges look like in a normal individual. But this is how a inflamed and infected meninges look like. So meninges can be infected by bacteria, leading to bacterial meningitis like Streptococcus, Neisseria, or Listeria as well. There could be viral meningitis, Coxsackie virus, herpes virus, HIV virus can infect the meninges. There could be fungal as well as parasitic meningitis as well. So all these things lead to an inflammation of the meninges, which might have overall effect on the brain functionality. Now the question is, which type of cells take part in the neuroinflammation? So how does neuroinflammation hampers the overall brain function? So our brain contains different type of cells apart from the neurons, including glial cells like astrocytes, oligodendrocytes, and microglia. All of these glial cells talk to each other, and they also talk to the neurons, and thereby creating a glial and neuronal interface. So these glial cells are heavily affected in neuroinflammation. And scientists think that many of these glial cells, including microglia and astrocytes, are actually the driver for neuroinflammation. So key focus has to be given on microglia and astrocytes. So microglia has diverse set of roles. Not every role is associated with neuroinflammation, but diverse roles include eliminating damaged neurons, synaptic pruning, release of reactive oxygen species, release of inflammatory cytokines, recruiting oligodendrocyte progenitor cells, promote neuronal repair, and even phagocytosize debris of foreign materials, or let's say a pathogen when it invades the brain. So thereby, it's really important in context of overall brain function. Microglia always survey the cellular environment. If you look at a resting microglia, one can see the processes are approaching and retracting several times as if they are surveilling. So they are looking for some invaders. So the surveillance mechanism is pretty prevalent in terms of microglia. So microglia are dynamic. They are like security guards, always monitoring for local environmental changes. Microglia can also work like scavengers. They engulf cellular debris. They engulf damaged portion of the neurons as well. So microglia has specific receptors known as pattern recognition receptors, or which recognize damage associated molecular pattern or pathogen associated molecular pattern, and thereby it can engulf these damaged debris or even entire pathogen and preventing them to harm the brain. Now, one of the key functions of microglia is basically synaptic pruning. So the way microglia affects the brain is altering the synapses, thereby it has the potential to alter the brain function. Microglial activation due to brain injury and infection is an important aspect. So whenever there is an injury in the brain, microglia converts itself from a resting state to an activated microglial state. Activated microglia secrete several cytokines which are pro-inflammatory in nature, which attracts many other cells, including T cells. So how does microglia know which is the injury site? It turns out the injury site secretes ATP, which is recognized by the purinergic receptor present on the microglia, and thereby they can reach the site of injury and evoke inflammation. Sometimes this neuroinflammation is for the good for the brain, but sometimes the chronic level or beyond control level of inflammation is bad for the brain, and that's what happens in many disease. Now microglia are diverse. There are different types of microglia. Each of these microglia has their own molecular marker and own function. So there are specific microglia which are discovered, which are heavily associated with diseases. They are known as activated microglia or disease associated microglia. One such evidence comes from Alzheimer's disease. Many of the Alzheimer's risk genes are actually enriched in microglia, such as TREM2 or MSA4. So that is why people think that microglia could be also a driver for neurodegenerative disorders. Anyway, this hypothesis is still debatable. Anyway, in Alzheimer's disease, the microglia engulfs many of these plaques and they becomes reactive. They secrete cytokines like interleukin 1, 6, TNF alpha, all which are pro-inflammatory in nature. They interact with astrocytes with these cytokines and make the astrocytes reactive. Astrocytes further can affect the neuron and synaptic properties. Thereby, it creates a vicious cycle in the Alzheimer's brain. Now, in Alzheimer's brain, people have identified a specific subcategory of microglia known as disease-associated microglia. But how these microglia are generated and how they are different, it turns out they downregulate microglial checkpoint genes and they have upregulated phagocytotic and lipid metabolism gene. Still, scientists are trying to figure out how they are beneficial or detrimental for the Alzheimer's brain. Now, when it comes to neuroinflammation and disease, multiple sclerosis is one of the disease which is heavily associated with neuroinflammation. So it leads to, in this disease, there is a progressive destruction of the myelin sheath around the axon, which leads to plethora of symptoms, and that gets worsened over time. So there are several cells like oligodendrocytes which are helping in the myelination process in the brain. And oligodendrocyte has unique proteins known as myelin basic protein, proteolipid protein, and myelin oligodendrocyte glycoprotein. All of these are pretty native to oligodendrocyte. So they are not supposed to be recognized as foreign. But due to an immune malfunction in multiple sclerosis, these proteins are detected or recognized as foreign entities. And thereby, autoreactive T cells are generated, which further activates B cells, which differentiates into plasma cells, which secretes antibody against these oligodendrocyte proteins, and thereby damaging the entire myelin sheath. And this is the key cause that evokes inflammation in these in the brain and the spinal cord of multiple sclerosis patient. It is also found that not only these autoreactive T cells harm the brain in this way, but also they differentiate into other subtypes such as Th1 and Th17. Th17 is one of the T cell that produces interleukin 17, which is a pro-inflammatory cytokine. And people are still debating whether these are the key cell types that can evoke inflammation in context of multiple sclerosis. Anyway, there are cells that can suppress autoreactive T cells known as T regulatory cells. And in case of multiple sclerosis, there is a dysregulation of these T regulatory cells, which cannot prevent the excessive activation of autoreactive T cells. That ultimately leads to inflammation. Not only multiple sclerosis, in context of Parkinson's disease, the neuroinflammation is pretty predominant. It has been seen microglial activation and microglial morphology alteration in the autopsy of the Parkinson's disease patients. Now, when it comes to amyotrophic lateral sclerosis, neuroinflammation is involved because reactive astrocytes and microglia are found in the nervous system of ALS patients. So, when we took the example of these disease, throughout this disease, we can understand that there are reactive astrocytes and microglia which can lead to neuroinflammation. That brings us to that idea that not only cells, cellular states can be an important driver of disease progression. Maybe astrocytes and microglia are good in their ground state and they are required for the brain activity. But when they are forced or pushed to become reactive, then they become harmful. Like you are normally cool or chilled in your normal state, but when somebody agitates you, you become agitated and you become furious. So this is a nice analogy that can tell you not only glial cells are important in context of neuroinflammation, the reactive states and their specific subsets are really important in that context. Also, neuroinflammation is associated with encephalitis, which is associated with viral infection, but microglial activation is a hallmark in this particular disease as well. So we understood and appreciate the common theme, that is microglial activation as a driver for neuroinflammation. So I hope this video was useful. If you like this video, give it a quick thumbs up. Don't forget to like, share and subscribe, and see you in the next video.